from tomorrow is the last day of the 9 weeks, and then you get a Friday off and a week off, so that's always nice. Today is the 24th, and so we are right here. A lot of you guys are already getting your labs back in the back. We'll wrap up organelles, and then next time I see you guys on Friday, we'll take our test. And our test, I think I said this last time, but our test is the last thing you have to do for this 9 weeks. So after I get your test grades in there, if you like your grade, great. If you're close and you want to up it a little bit and want to turn in some voluntary homework, cool. If you do some voluntary homework here and you're like, I don't really want to turn it in on this 9 weeks because I like my grade, can I turn it in next 9 weeks? Yes. If you have voluntary homework that you want to go on next 9 weeks, though, don't give it to me this 9 weeks because that is confusing. So if you hand it to me before the 9 weeks is over, I assume you want it on this one. And if you hand it to me like October 20 whatever, whenever we come back, then I'll put it on next 9 weeks. So even if it's like Unit 2 stuff, getting ready for this test, if you don't want to turn it in until next 9 weeks, then that's totally fine. But just don't give it to me early and then like write a little note about like count it later, because I'll probably end up accidentally counting it now. On the 8th is the PSAT. So that means if you're a senior and you're not taking the TSIA thing or whatever, then you'll get to sleep in. I haven't seen the schedule yet, but isn't it normally like 11:30, 11:45, something like that? They do take roll, so make sure you do show up, and they probably scan your badge or whatever they do. I don't know, used to, we just had this giant list and we would highlight that you're here. So that'll be kind of a weird day. And if you're a junior, then good luck. It counts for like national merit stuff in 11th grade. And there are not a lot of colleges, but there are a few colleges that are just obsessed with wanting to be able to say they have more national merit semifinalists than anybody else. And so the best academic scholarships in the country are national merit semifinalist scholarships. So semifinalist and finalist. UT Dallas is a full ride if you're a finalist, plus $4,000 for study abroad. It covers room, board, tuition, money for a computer, books, parking. You're like, how much is parking? Do any of y'all have friends that go to UT Austin or any place like that? About $1,000 a semester. Arkansas University. I have a family friend. His daughter's currently at Arkansas, and parking is $1,000 per semester, and on game day, you can't be on campus or they give you a ticket. So on game day, you have to leave, and where do you park? It's just like not here, and then you still pay $1,000 a semester to park. Imagine if you go to some place like New York. I don't know what parking is in New York, but yeah. So ACT, colleges, you can still get good scholarships. So like doing well on the SAT and ACT helps, but there is, to my knowledge, no like just full ride scholarships. So usually for doing great on the SAT and ACT and stuff, you can get your tuition waived. So at UT Dallas, tuition is like $15,000, and then room, board, tuition, all that stuff is about $30,000. And so if I got like a perfect ACT, and I was, you know, #1 in my class or whatever, I can probably get my tuition waived. But on the pre-SAT, if I score on the top about 0.5%, everything's free. So it's weird. You would have thought that the pre-SAT was less important than the SAT. Kind of like how the scrimmage should be less important than the real game, but it's not. Now on the PSAT, if I'm not national merit and I do great, they generally give you nothing. Whereas if I do really well on the SAT, then I end up getting scholarships. So the PSAT is kind of all or nothing. But there's a few places. Alabama's free. OU used to be free. Tulsa University's free if you're a national merit semifinalist. So sleep well and good luck. Any questions about our schedule? Cool. Next class is our test. The test is a little bit more heavy on membranes and transport versus organelles. It's probably 60-40. I didn't sit down and tally it up, but I'm just looking at it and I'm like, yeah, that's about 60-40, which makes sense because we spent probably 60-40 class time on this stuff versus organelles. So we also spent more time on membranes and transport than we will organelles. So that makes sense. Number of questions is about the same. I can't remember. I think it came out to like 33 or something, and one 10-point free response. So our first test, I think, was like 35 and a 10-point free response. So it's about the same length. All right. Any questions on that stuff? Cool. All right. Organelles then. All right. So remind me, why did I need a nucleus in eukaryotic cells? Yeah, I'm protecting my DNA from who? Yeah, DNA. So there are DNA enzymes that can destroy DNA, but they can also destroy my DNA. So I want to protect my DNA from DNase. Why have DNase in there to begin with? Yeah, viruses. DNA-based viruses can get into my cells, and so my counterattack is DNase, but then DNase could also attack my DNA, so store my DNA in the nucleus. So basically, DNA should never be outside of the nucleus. The only way it can be outside of the nucleus and not get wrecked is if I go and I disable and turn off all the DNase enzymes, which I can do when I'm doing cell division. But when I'm doing that, I'm now like extra at risk of getting invaded by a virus. Cool. RNA can leave the nucleus, and it needs to because a lot of the proteins that I make are huge. So RNA successfully leaves the nucleus without getting destroyed by DNase. That's why it's not called RNase. So RNA leaks out, and a ribosome lands on it and starts building protein from it because ribosomes read RNA and build protein. And then if I want to package that protein, if that protein is going to like go to work right here in the cell, then it lets go of it and that protein goes to work right here in the cell. But if I have to ship it out of the cell, then I need to package it. It's kind of like if I was going to send somebody this, if they live in another state, I need to package it, kind of like the analogy I gave last class. I'd like put it in something hollow, put an address on it, and send it to my friend in Oklahoma. But if like Hacilius next door needs this, I don't go and like, you know, Jessica Hacilius at wherever she lives and then like put it in the mail. I just kind of like walk it over there. So if I build this protein right here with a ribosome and that protein is going to like do a job out here, then I don't do all this packaging crap. But if I am going to ship it out, I package it. So I shove it into the hollow sole of the ER. That ER pinches off. And that little like kind of package is called a vesicle. Kinesin will carry it, take it to Golgi. What was it that Golgi was doing by adding these chemical tags? Yeah, it's labeling. And what good does that label do for me? How does putting a witch's hat on there help at all? Yeah, Theo. Yeah, it helps the receptor identify it. So if I dump this chemical, this purple protein, whatever it is, into my bloodstream, and it has this little like chemical tag on there, then eventually, as it's cruising through my bloodstream, it might make it to that cell. And if that cell has a receptor for that hat, then that cell responds to this chemical. If that cell doesn't have that receptor, then that cell doesn't respond. So not every cell in my body needs to respond to adrenaline. Not every cell in my body needs to respond to testosterone, estrogen, growth hormone, aldosterone, whatever the heck, insulin. Actually, all cells in my body respond to insulin. But if I don't want a cell to respond, then I don't make receptors for it. So anytime I'm having like hormonal problems, like if I have to see an endocrinologist, they're like, hey, something's wrong with my metabolism, I got to go to an endocrinologist. Endocrinologists study like hormones and hormone systems. So if something's wrong with the way that the system is working, it's usually either I don't make the hormone correctly, so this guy is incorrect, or maybe this guy is correct, but the receptors are incorrect or they aren't there. So in type 2 diabetes, for example, I'm eating a crappy diet typically. I've got on way too much body fat. And so my body is constantly making insulin to try to control my blood sugar. But if levels of insulin get too high and I'm a type 2 diabetic, my cells quit making receptors. So if you've ever heard of like being insulin insensitive or this person needs better insulin sensitivity. The way that I'm insulin insensitive is like, hey, no matter how much insulin I dump into my bloodstream, if I'm not making very many receptors for it, it doesn't matter. No matter how many quarterbacks I have throwing the ball into the end zone, if I've got 50 quarterbacks but I've only got 2 wide receivers, I'm limited. So this is like throwing the ball. The receptors are like catching it. You need to have like a somewhat like logical number, otherwise it's not going to work, right? Right about there pretty much catches us up to where we've been. Let me see. I think rain's gone. So real quick for the KCal crew, before you leave, if you want your lab, they're in the out folder back there. You guys have been turning stuff into the green folders. They're folders that say out. You take stuff out of the out folders. So if you find your out folders back there, if you want your lab, you can grab it. And then next class, for you KCal crew, is our test, because we're right here. And then on our test, it's more heavy on membranes and transport than organelles. So organelles is like kind of this stuff. Anything that started with the membrane, that's more like membrane transport. It's probably, instead of 50-50, it's probably 60-40, a little bit more membranes and transport. So if you're like, what should I study? I'd start with that stuff, unless I've like got that crap locked down because I studied hard for the quiz or something, and now I study organelles. But there's more of this stuff than there is organelles. And this stuff is harder than the organelle stuff too. So, yeah. All right. So now let's add in a few new things. So I've got my cell up here, and I've got my ER, Golgi. Great. I've got 2 versions of ER. I've got rough and smooth. Did we talk at all about rough and smooth? What made rough ER rough? Ribosomes, yeah. So I might have rough ER and ribosomes are landing on there. Whatever's coming out of rough ER is going to be protein because ribosomes are shoving protein in there. And then smooth ER is if I'm packaging anything else other than protein. So if I had a steroid hormone, a cholesterol-based hormone like testosterone, aldosterone, you're like, well, that's not a protein, so ribosomes aren't making it. So those are being transported by like smooth ER. Smooth ER can also just hold a bunch of enzymes that are teeny tiny, so I don't notice them. That's why it doesn't look rough, but I'm putting tiny blue dots on there that most of you guys can't see because they're too small. So that's smooth ER. Sir. This is rough ER. Rrr. All right, so I package that stuff. I send it to Golgi, put a tag on there. Great. If I'm following along in my notes, that's not your notes. I might just have to ask somebody what's next. Oh, here's some. So if I'm following along in my notes, if I go to page 4, nucleus organelle starts at the bottom. Then there's ER, then ribosomes, then Golgi. All right, next up, lysosomes. Lysosomes look super boring, but they're actually really interesting. So lysosomes. If I'm looking at a cell, then a lysosome, like I said, looks kind of dull. A lysosome might look like this. Just like a little sphere. And it's got enzymes in there. And you might recall from your Biology 1 class, your teacher probably said something like lysosomes are like the custodian of the cell. They sort of break down trash. So I might have waste material in my cells. They might break that down. I might bring something in through pinocytosis. Remember how with pinocytosis, pretend that there's like a bunch of dots out here, purple dots, blue dots, green dots, whatever. I might bring it all in, and maybe I only want the green dots. Well then, if I need to like break down the purple dots, lysosomes can help me do that. If a mitochondrion is getting pretty old, they're volatile. They're kind of like batteries, so they kind of like age out fast. If I have a really old mitochondria, lysosome can kind of break them down. So whether it's actual trash or it's just like an old organelle, they can just sort of like dispose of stuff. So lysosomes, you can kind of think of them as like little bitty stomachs. They've got all these enzymes in there that are just good at digesting general stuff. So they have enzymes that can break down lipids, enzymes that can break down proteins, enzymes that can break down carbs. Again, much like my stomach, My stomach is pretty good at chemically breaking down everything from pizza to hamburgers to a sugary snack, whatever. So that's kind of what lysosomes are like. So here's how it's supposed to go. Let's say I've got a bacteria out here. There he is. He's going to die. We kind of saw this earlier. This was phagocytosis. So here's this bacterium. Bacterium is one, singular. Bacteria is plural. So here's this bacterium, and we're going to phagocytize him. There he is. So this phospholipid bilayer just kind of like pinches inward, like that. And so eventually it's like a bubble. Again, kind of like a lava lamp bubble. And then what's supposed to happen is now he's on the inside, but I've got to kill him. And so a whole bunch of lysosomes like surround him. Okay, so I'm going to draw one big lysosome up here so you can kind of see what's going on. But they wouldn't be outside the cell. They're all inside the cell. So they're going to surround it. Now, right now, that guy is inactive. They might call it inactive versus active. Some books call it primary versus secondary, which is super dumb. Just call it inactive and active. That way, like, based on the name, I immediately know what it means. I think your book says inactive and active, if I remember right. Some books, again, say primary and secondary. So primary is inactive because that's first. Secondary, it can become activated. All right, so here's the deal. I've got all these enzymes in here, and they're really good at breaking down stuff. All right, cool. But it turns out those enzymes work at low pH. So I'm just going to make up a low pH. They work great at like pH 2. It might be 1.5, it might be 3, but it's like a low pH. All right, cool. So a low pH, a pH less than 7. Is a pH less than 7, is that more acidic or is that more basic? That means it's acidic. Acids are lower than 7, bases are above 7. It always feels like higher pH numbers should be acids, but they're not. So that means it's really acidic, kind of like my stomach. Okay, the reason my stomach pH is so low is because of something pretty genius. So I've got my stomach. It's basically just like a muscular container. And the reason I have stomach acid is stomach acid doesn't actually break down anything very well. If I check the pH of my stomach acid, it's about pH 2. That is true. That's not a guess like this guy is. It's about pH 2. If I take a piece of chicken and I take hydrochloric acid, that's the acid that's in my stomach. I secrete hydrochloric acid in there. If I take a piece of chicken and I put it in a vat of hydrochloric acid on Friday, and I come back and check it on Monday, it is unchanged. So 2 pH hydrochloric acid doesn't break down chicken. It'll look a little bit fuzzy on the side, kind of like if I just left it in water, it would look like the outside is kind of like gross looking, but it's not like it would dissolve. So you might be like, well, hey, why do I need stomach acid? It turns out the enzymes in my stomach only work at really low pHs. So imagine this is my stomach. I'm going to erase this in a second. But imagine this is my stomach, and these enzymes work at a pH 2. But right now, inside, it's like, I don't know, a pH of 7. They need a pH of 2. So are those enzymes currently on? No. So as long as I can keep them from denaturing, as long as they're just sort of like change shape enough to where they don't work, but then if I change the pH to 2, they pop back into shape and they work, everything's great. This is a tough question. Talk with somebody around you. Why is this a good system? Why not just have enzymes in my stomach that work great at pH 7? My stomach is pH 7, and so now the enzymes are basically always able to break down food. Why is it that my enzymes only work well at pH 2, and so I have to like drop the pH in my stomach? Can anybody think of a good reason why that is? Why do it this way? It's a hard question. I'll give you a hint, it's safety related. All right. You haven't had much time. Anybody want to take a crack at it? It's a hard one. Okay, so my stomach doesn't eat itself. So one thing is like, hey, my stomach has pepsin in there. It's good at breaking peptide bonds. It should be called peptase. It's not. It's called pepsin. So it's a protein. We named it a protein, pepsin, and then later on they figured out it was an enzyme. So pepsin's great at breaking down protein, muscle. But my stomach is made out of protein, muscle. In fact, I can like eat stomach. So I can eat like cow stomach if I want. So my stomach can be digesting stomach. Think about that. So I can do that. Well, what's keeping my stomach from digesting itself is I have this layer of snot around the lining of my stomach. That's why it doesn't eat itself. So it's not that. That's a very good idea, though. It's something different. It's related to that, though. You're like in the right ballpark. Okay, where do you think the pH is outside of my stomach? Just like kind of in my abdomen. Do you think it's super acidic, or do you think it's kind of more like probably closer to water, maybe 7-ish or something? Yeah, probably 7-ish. I don't know what it is. Might be 6.5, might be 7.5. I know my blood is like 7.35 to 7.45. So in my abdomen, let's say it's 7. Let's say I get in a car wreck and my stomach ruptures. Inside my stomach was pH 2 because I just ate some pizza. And I get in a car wreck, it ruptures. So it was pH 2. Those enzymes were like breaking down pizza. My stomach just ruptures. My stomach sits right here, right on top of my liver. Is it possible for me to eat and digest liver? Like, can I buy liver at the store and fry it up and eat it? Yeah, if I'm gross. So my dad's from Tennessee. He eats crap like liver. They're like, does that filter everything out of your body? It's like, yeah. And it's like, I think I want to eat that. And they're like, it's high in iron. You know what else is high in iron? Cereal. So eat cereal. Don't eat liver. Anyway, so I get in a wreck. My stomach ruptures. What keeps those enzymes from just like digesting my own liver? The difference in pH. As soon as my stomach ruptures and those enzymes that work great at pH 2 leak out of my stomach and hit pH 7, aren't they immediately disabled? So I can't eat myself. So if my stomach leaks digestive enzymes into my abdomen, they're immediately turned off. That's why your stomach is set up the way that it is. Okay, now, you're like, okay, I thought we were talking about lysosomes. Exact same idea with lysosomes. Let's say that I bump into something really hard and some lysosomes in my cells burst. Won't they just leak out enzymes everywhere? And these guys are good at breaking down things like carbs and lipids and proteins. But I'm carbs and lipids and proteins. So if they leak out, they immediately are disabled because they only work at pH 2. And out here, it's not pH 2. So as soon as my lysosomes rupture, all these guys get disabled before they can go and like digest myself. So that's actually pretty clever. So these inactive lysosomes will all surround, they'll all surround this bacteria that I just gobbled up. And then what they do is this. They have these proton pumps on them. And a proton is H+, hydrogen ions. They will start pumping in hydrogen ions, and that drops the pH. The capital H and pH is hydrogen ions. So whenever they're going to start working, let's say the pH in there right now is like pH 6 or something. And all of a sudden they like have to get busy and go to work. We have to drop the pH to 2. How do we do that? We just start pumping in H+. And so now my pH is 5, 4. And then eventually my pH is 2. And so now those enzymes are active and they're ready to do some killing. So I have lysosomes surround this bacteria that I just gobbled up. They'll start pumping in hydrogen ions to become active, and then that is phospholipid bilayer. That is phospholipid bilayer. So if they bump into each other, then now, isn't this guy just like getting covered with all these enzymes? Yeah. And so that's how I break down all the trash. So lysosomes are inactive. If they bump into something and they're going to start trying to like destroy it, then they'll turn on these hydrogen ion pumps, proton pumps. That'll drop the pH. Now they become active and they can do some damage. If something happens and I hit the wall really hard and this all pops, then those enzymes immediately turn off because the pH out here is not 2. It's pretty clever. Does that make sense? So it works just like my stomach does. Right now, let's say that you're a clever bacteria. Hang on one second. Let me grab a picture really fast. How many of you guys, I don't think we've talked about it in this class yet. Do you guys remember a few years ago, maybe 5 years ago, when you couldn't buy Bluebell ice cream for about a year? Bluebell ice cream was pulled from the shelves. Yeah. Have I told you guys about this? Okay. So they pulled it from shelves. Do you remember what bacteria? It wasn't E. coli. It's almost always E. coli. That's why we always end up pulling romaine lettuce about every 3 years. There's no Caesar salad because they get poop on the lettuce. Apparently we get all of our romaine lettuce from like 2 or 3 farms somewhere, and somebody gets like poop all over it. And so they'll be like, no romaine lettuce. And you're like, isn't it okay to eat? And you're like, it's got poop on it. And then like a few years later, like happens again. I'm like, hey, move that pipe over there. Like, what are you doing? So anyway, it turns out it was due to listeria infections. So listeria bacteria, tell me that shouldn't be some song lyrics. It just rhymes great. So listeria bacteria works like this. So usually the way that viruses make me sick is they invade my cells start copying their genetic material, and my cells become a virus factory. Bacteria, the way that they make it sick, how'd it go? Oh, okay. So with the bacteria, the way that they normally make me sick is like pretend that you're all eukaryotic cells in a person. You're all Mr. Cotton cells. I'm a bacteria. I just walk in here and I just start oozing out something that kills you. And that's how I harm you. I never invade cells. So if I'm strep, then I end up on the back of your throat somewhere and I just start oozing stuff that kills cells. And that's why strep is like destroying cells. Listeria is different though. Listeria is a clever bacteria. So here is listeria. He doesn't have a frowny face because he wants to be captured. So here's my cell. My cell's like, there's a bacteria. I'm going to envelop this guy. And listeria is like, ha-ha. There you go. So my cell will phagocytize him. However, he was wanting to get inside the cell in the first place. Y'all have heard the whole like Trojan horse thing? Pretend that there's like just us. There's like 32 of us, 33 with me, and we're trying to like conquer this castle over here. If the walls are too high, they got 40-foot tall walls. There's like 5,000 soldiers in there. We can't compete. We live in like a tent. They live in like this, again, 40-foot tall walls, castle, all that. We get close, they dump boiling oil on us. It's horrible. So we're like, all right, here's what we'll do. A few of us will get captured on purpose. We'll go inside. Once we're inside, before they take us to jail, maybe if we can bust out, then we can like slaughter everybody. So that's what this bacteria does. He gets engulfed on purpose, But before the lysosomes can surround him and digest him, he breaks free. And now he wreaks havoc. So he'll reproduce and just start destroying the cell from the inside out. So he acts a lot more like a virus. So that's listeria. And that's what Bluebell was infected with 5, 6, 7 years ago, whatever that was. So here's a cell, a white blood cell, and its job is to go around and like gobble up bacteria if it sees them. I think the narrator on this one's pretty good if I remember right. So we'll keep the narrator on.
These bacteria move throughout the cytosol by recruiting host cell actin, which polymerizes and pushes them forward, producing a convex tail in the Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path.
So now imagine if I have a listeria infection, then, Cool, thank you. Yeah, thank you. Like a ninja. So imagine I'm told like I have a listeria infection. And you're like, well, what does that mean? And it's like, well, there's like bacteria inside of your cells. And I'm like, are they like flying around all creepy, crawly, and weird? And they're like, hell yeah. And I'm like, that sounds weird. And then they're like, oh, by the way, they're also reproducing inside of your cells. So if you didn't already feel violated.
Closely, we can see a bacterium divide inside the host cell. Immediately after separation, the two daughters...
There you go. One bacteria just divided into two. So that's why you didn't have Bluebell ice cream for a while, listeria. The other common place that you would get like a listeria infection, I even saw it 2 days ago, is like cold cuts. So obviously if I have like any other kind of meat, if I get like fresh ground beef or whatever, that's good for like a few days if I just keep it in the fridge. But they'll like slice up some turkey or whatever, and that stays for longer. It's got a lot more preservatives in it. That's why it's considered less healthy for me. Like anything that lasts a long time has to have a lot of preservatives in it, right? If you get the fresh bread, you know how they have like regular bread, like Wonder Bread, loaf bread, and that might last like a few weeks before it gets nasty. But then they also have like the fancy bread part where they have those like big old loaves of French bread. And you notice how if you buy the French bread in like 3 days it goes bad? That's because it doesn't have a lot of preservatives. That's why it doesn't preserve very well. So in general, if you have something and you're able to store it for like a really, really long time, it probably has more preservatives in it. Otherwise, it should go nasty faster. If I have a banana, it goes nasty quicker than a Twinkie, right? I can have some Twinkie or a cupcake, and you're like, oh, how old's that? And you're like, doesn't matter. Just open it up. It's fine. And you're like, oh, I've heard there's a lot of preservatives in this. Yeah, that cupcake's been there for like a month, and it's fine. Oh, doesn't it have like whipped cream in it? Isn't it unrefrigerated? And they're like, yeah. And you're like, milk unrefrigerated goes bad by like tomorrow. And you're like, not this milk. This milk's good until November the 15th. And you're like, oh, this is probably full of preservatives, right? And you're like, I think you're catching on, yeah. So anyway, listeria, if there's any outbreaks, it's usually like sliced deli meat, and then apparently Bluebell ice cream. So, yeah. Okay, so lysosomes. Next, after lysosomes, peroxisomes. Peroxisomes I'm just going to mention just because you might go into college. They're basically like lysosomes. So the likelihood that you see a peroxisome on the AP Bio test is like slim, unless they're looking for, we need a really hard question that few people know so that only the fives get it. But peroxisomes are kind of like lysosomes, except the chemical that they use inside is hydrogen peroxide. So they use hydrogen peroxide to break down stuff. Hydrogen peroxide is great at breaking down stuff. Hydrogen peroxide is great at killing stuff. About 5 years ago, when some of you guys were learning at home and some of you guys were here and we were all wearing masks, and your teachers had to like spray down your desks between every single class. We were spraying it down with stuff where the active ingredient was hydrogen peroxide, because hydrogen peroxide is great at destroying everything. So even my clothes, if I get hydrogen peroxide on my shirt, it's going to like bleach out my shirt and there'll be a hole in there. All right, so peroxisomes, don't worry about it. Below that, lipid droplets, that will not be on the AP Bio test. I was just kind of pointing out that in 2005, they discovered another organelle. Like organelles are things in a cell that we can see under a microscope. And in 2005, they discovered lipid droplets, a new organelle. That'd be like if we found out that there was like a planet between us and Mars. They're like, wait, I thought we were working on like other galaxies and how many of them have like planet systems like ours. It's like, oh yeah, turns out there's something between Earth and Mars. You're like, how do you, I thought we had this solar system like knocked out like decades ago. So they're still discovering new organelles like lipid droplets. Do what? Okay, anyway. A lot of controversy on lipid droplets. Okay, the next one, mitochondria and then chloroplasts. So mitochondria and chloroplasts, we spend basically all of our next unit on how do mitochondria work, how do chloroplasts work. But for now, we're going to look at the basics. Mitochondria is super interesting, and the stuff that we learn about it, even just today, before we get into a lot of details, is super interesting. So if I'm drawing a mitochondrion, mitochondrion is one of them. Mitochondria is plural. If I'm drawing a mitochondrion, it just looks like a kidney bean maybe. Some of them are really long, so they're more like kind of hot dog shaped. Some are shorter and they're almost like just perfect spheres. So it's kind of like all the different versions from spheres to like elongated, but most of them are pretty elongated. So they aren't really like perfect spheres usually. They're almost always like pretty elongated. I got some pictures of them. Let's see. There's your mitochondria. Here's your mitochondria underneath a microscope. And if we cut them open, we notice something interesting. The inside layer of the mitochondria is all folded. So it's got a phospholipid bilayer right here. That black line is all phospholipid bilayer. But then the inner membrane is also phospholipid bilayer, and it looks like this. So the inner membrane is also phospholipid bilayer, and it looks all squiggly like that. So if I was trying to get from outside the mitochondria to like the very middle, I would go through a phospholipid bilayer, the black one, then I go through another phospholipid bilayer, the kind of aqua one. All right, that becomes important just a little bit later. What's the job of mitochondria? Yeah, I'm trying to manufacture ATP. So I kind of think of it as a converter. It's converting food energy into ATP energy. It's not really creating energy. Physics says I can't create or destroy energy. I'm just converting one type to another. So mitochondria eventually is able to convert cheeseburger energy into ATP. And that's important because my cells run almost exclusively on ATP. So the analogy I tell my Bio 1 students is Timber Creek. Anything in Timber Creek that's running on energy, isn't it pretty much electricity? Like that, batteries, but still electricity, that. With the exception of the parking lot where we've got gasoline, pretty much everything at Timber Creek that runs on energy runs on electricity. We might have natural gas-powered hot water heaters or something in the cafeteria, but it's almost exclusively electricity. All right. Does a piece of pizza have energy in it? Yeah. We might say it has 500 calories of energy in it. Can I, well, not can I can do anything. Should I shove a piece of pizza into like the grate of that projector? Will that help power it? No, because that projector only runs on electricity. If I could somehow convert pizza energy into electricity, that would work. Does the sunlight have energy? Sure. if I take my projector and I just put it out in the sun, does it charge up? No. But if I had a solar panel and I could convert sunlight energy into electricity, then I could power that. That's what mitochondria is doing for my cells. My cells run almost exclusively on ATP. We'll learn a couple of exceptions this year, but it runs almost exclusively on ATP, so it's converting whatever I'm eating into ATP. If I decide to eat plastic cups, is that food? Nope. Why? Because my mitochondria cannot convert it into ATP, so it's not food, and now I just look like that kid that's eating plastic cups. And you're like, are there kids that eat plastic cups? Yes. So let me pause the video before I tell you this. And we're back. So we got mitochondria. The inner membrane's all folded. Anytime something's all folded in biology, it's almost always for the same reason. Why is the outside of my brain folded, the inside of my intestines? When something's heavily folded, why is that usually? Yeah, more surface area. So here's the thing. I have these chemicals. We learn more about them in the very next unit. But I have these enzymes that make ATP. Does anybody happen to remember what they were called? If you were in honors bio, you should have learned about them. If you were in on-level bio, you were drinking the Gatorade. Yeah, I'm not saying you were. You might be like, I was in on-level bio. You were the 80% of the class that was cool. So most of my on-level bio kids were great, but it's just that 20%. Anybody remember what that was called? It's an enzyme, so what does it probably end in? ACE, and it makes ATP, so it was called ATP synthase. It's an enzyme, ACE, and it synthesizes ATP. We've got an enzyme, ATPase, that breaks down ATP, but ATP synthase synthesizes. Synthesize means to make. You might have heard that some chemical is man-made, it's synthetic. So synth means to make. Back in the day, we didn't call keyboards keyboards, we called them synthesizers because they were making all kinds of new weird sounds. So ATP synthase. We'll see why in the next unit, but trust me for now, ATP synthase only works if it's stuck in this membrane. So if there's an ATP synthase just floating around out here, it doesn't work at all. It can't do its job. I compare it to like a window opening. Window openings only work if there's a wall. Right? If I go and stand in the middle of the parking lot and I'm like, here's my window opening, you're like, were you drinking the Kool-Aid? Were you drinking the Gatorade? And I was like, sometimes. It only works if there's a wall. So if there's a wall and all of a sudden I like cut out a part of it and I'm like, here's the window opening, you're like, that makes sense. So for now, just trust me, those free-floating ATP synthases won't work. They have to be stuck in a membrane. All right, cool. So my students will tell me that I increase the surface area by folding stuff. And that is true. That is completely correct. But I want to show you kind of like what that looks like if you're having trouble like understanding what all that means. Let's see. My ATP synthases are orange. All right, so if the job of ATP synthase is to make ATP, the more ATP synthases I have, the faster I can make ATP, right? If I run a restaurant, I've got 10 employees making tacos for Taco Tuesday, and I need to make twice as many tacos. It's like, all right, well, instead of having 2 people, I need to have 20 people, and I should be able to make twice as much stuff. So with this, pretend that all these orange dots are these orange dots ATP synthase. Maybe I can fit 100 orange dots on here. This is the inner membrane, the folded one. My pocket is the outer membrane, the black one. So I can fit this in my pocket. There you go. I can fit that many ATP synthases in my pocket. But let's say that that's not enough to keep me alive. So instead, I need to cover a piece of paper this big with orange dots. That's probably 12 times the surface area. It's probably 4 by 3 of these. So if this is 12 times the size, shouldn't it be able to hold like 12 times as many dots? Yep. But does this fit as is in my pocket very well? No. So what do? very carefully fold it, and now, there you go. It's in my pocket. I just fit an 8.5 by 11 sheet of paper into like my 4 by 4 inch pocket. At first that seems impossible, but it's not. I just folded it. So that's what we mean by increasing surface area. If I fold stuff, I can fit something this big into a space this big. That means more ATP synthase, more ATP per second. Mitochondria. Cool. Some scientists were looking at mitochondria. And what I'm about to tell you is not fringe science. This is textbook science. So fringe science would be something that they discovered in the last 6 months. They've done a few studies. Seems promising. It's like, hey, here's what we figured out. Now a bunch of other scientists need to do studies to kind of confirm that it's true. There's a study that came out yesterday that's suggesting a link between Tylenol consumption in pregnant mothers and autism. But before we go and say, hey, Tylenol in pregnant moms is causing autism, it's like, oh, there's a correlation, but like this came out like, again, I think yesterday it was published. Now we need a bunch of other scientists to do a bunch of studies and see like, hey, do we have like dozens and dozens of studies that are like, yeah, there seems to be a pretty close link? Or is it like, no, that was kind of a one-off? So fringe science is usually new. What I'm going to tell you about is textbook science. So if you have 50 cell biologists in a room, all 50 will agree with this. Okay, so some scientists were looking at mitochondria, and they noticed something. Let's say that Bill is talking to another scientist named Bill. He's like, Bill. He's like, yeah, Bill, what's up? I noticed something. I was looking at mitochondria, as I often do, and I noticed inside they have their own DNA. And it's circular. So their DNA, it's still double helix, just like ours. It just happens to be in a circle. So let me show you what I mean. I know that you know like what a circle is. But here's my DNA. So our DNA is double helix like this, and it's just in these big long strands. We might crunch down those strands around histones and stuff to make a chromosome. With their DNA, everything's the same except this end and this end join. So now it's a circle. But otherwise, it's still just like regular DNA. The only difference is my mitochondria DNA looks like this. My nuclear DNA looks like this. So it turns out you have two sets of DNA inside of you. All right, cool. Then they were looking, and he's like, hey, Bill. And he's like, yeah, Bill. He noticed something else. He noticed something that looked like this. Looked like a pair of hamburger buns. What's that? Ribosomes. So they noticed mitochondria, all of our mitochondria, all of your mitochondria. This isn't like way back in the day in the evolution of humans. This is like 2025 humans. Our mitochondria have their own circular DNA inside, and they also have their own ribosomes. So they started looking at that, and Bill was like, hey, Bill, you know, I was also looking at bacteria the other day. And in a bacterium, on the inside, all they've got is DNA and ribosomes. Bacterial DNA is circular. In fact, the only circular DNA found in nature is in bacteria, as well as our mitochondria. And they have ribosomes, but their ribosomes are different from all eukaryotes. So their ribosomes do the exact same job, they're just different. Kind of like a Ford F-150 pickup truck and like a Dodge Ram, they're identifiably different. I'm like, that one's the Ford, that one's the Ram. But don't they basically do the same job? An iPhone versus a Samsung phone, they're different. I can identify them as like they're different. They run on, you know, this one uses this operating system, the I operating system, and this one uses some other one, but they do the same things. They both text, they both take pictures. Turns out there's two versions of ribosomes in the world. Bacteria have a version of ribosomes that are different from ours. The ribosomes that mitochondria have are the bacteria type. In fact, there's an antibiotic that you take. Let's say you have strep throat or something like that. You go to the doctor, they give you antibiotics. There's an antibiotic that works, bless you. There's an antibiotic that works by attacking bacteria ribosomes. Bacteria ribosomes are different than ours. So I can eat it safely, and it doesn't attack my own ribosomes. It will only attack bacteria ribosomes. And so that's how that antibiotic works at like destroying those bacteria. Turns out whenever I take that particular drug, some of it makes it through my membranes into my mitochondria, and it also attacks mitochondrial ribosomes. Why? Because, this is Mr. Scott. What's up? Yes, I will send him. Thanks. Bye. He responded with what's up? There you go, Logan. All right, so I notice that whenever I take this particular antibiotic, I don't remember the name of it, but it works by inhibiting ribosomes in bacteria, and that's why it kills bacteria. It doesn't harm my ribosomes because my ribosomes are different. So it's kind of like if I created a virus that's really good at attacking iPhone software, that should have no effect on Samsung phones. So then we started looking at a cell, and they're watching the mitochondria. Turns out mitochondria divide, like we saw earlier in that video. Mitochondria divide by this process called binary fission. You don't have to know anything about it, except that it's different than how we divide by mitosis. Turns out mitochondria divide by binary fission on their own in my cells. They don't divide by mitosis. They divide by binary fission, just like bacteria. Then they noticed mitochondria have two phospholipid bilayers. Very few things. ER, Golgi, those are all single layers. Lysosome, that's a single phospholipid bilayer. Here they have two phospholipid bilayers. So here's the theory on how mitochondria came to be. I had a cell way back in the day. It was a bacterium. It would have its own circular DNA, and it would have ribosomes. Let's say a cell tried to eat it. Why would we think a cell would try to eat it? That's what my modern immune cells do. But it wasn't successful. Now, wouldn't this thing have two phospholipid bilayers? The blue one and the black one? Wouldn't it have circular DNA and ribosomes, just like bacteria? That's how we think our ribosomes and DNA ended up in mitochondria. We think that our cells tried to gobble up a mitochondria, but were unsuccessful. So mitochondria came from bacteria. Again, this is not fringe science. This is textbook science. So this is not debated in the same way that like there aren't a bunch of scientists that are like DNA is a lie or something like that. Like, you know, big B, little b, that's a bunch of, that big pharma wanted you to, we invented Punnett squares and something like that. So it's kind of like, hey, we all accept that like DNA is genetic material and there's RNA and all that. So we think the mitochondria came from incompletely digested bacteria. Here's the logic behind it a little bit farther. Pretend that I walk up to you and I'm like, look, I invented a brand new thing. It looks like this. Here you go. Here's a brand new thing. Check it out. Isn't it awesome? And you'll be like, oh, let me see this. You just wrapped an expo pen in like a piece of paper, and I'd be like, nuh-uh. Gatorade. And I'd be like, yeah, you did. And they'd be like, prove it. And you'd be like, well, here's an expo pin. Do we agree this is an expo pin? He's like, yeah. I'd be like, here's your thing. Do you see how it says expo like that? And see how this says expo? And I'm like, coincidence? And he's like, well, an expo pin, like the top comes off. And on your thing, like the top comes off. Not strong enough, I guess. You work out my left hand expo pin cap remover. And then like an expo pin does this. And your thing does that. And so that's why I think that it's just an expo pin with like a membrane around it. And that's the argument of scientists. It's like remove that black membrane and you basically have a bacteria. Looks like a bacteria, has DNA like a bacteria, has ribosomes like a bacteria, responds to antibiotics like a bacteria, divides like a bacteria. We have nothing in there that's eukaryote-like. Everything, anytime that we learn something new about bacteria and we compare it to this, it's like, oh yes, it does that too. So that's where we think mitochondria came from. Isn't that super weird? Think they have like little bacteria dividing in me? Yeah.
Did they just like, the mitochondria just make the ATP like enzyme?
Yeah. So here's the thing, you might be like, okay, so in eukaryotic cells, before there was mitochondria, how did they make ATP? We talk about it more in the next unit, but glycolysis. Glycolysis doesn't require ATP. So mitochondria make ATP, but without mitochondria, I can do glycolysis. Inside of bacteria, they can do glycolysis. Inside of my own cells, I can do glycolysis. So bacteria do have some proteins that are similar to ATP synthase. They kind of do a version of cellular respiration. It's pretty different, but kind of. So we think that all the cellular respiration stuff evolved over time. That might be a hard pill to swallow, but if I'm basically describing, I've got this, I think it's just an expo pin wrapped up. If you're going to topple that theory, you're going to have to come up with a better explanation, which is really hard to do. Now, my explanation is over millions of years, they got really good at making ATP. And you're like, oh, that's a little bit far-fetched. Yeah, but like, this is not. So we think that mitochondria came from bacteria. Super weird. But yeah, it would have to evolve over time to get really good at making ATP. But I can buy that more than I can just buy that it's coincidence that it happens to look like bacteria. All right, that's mitochondria. If I look at chloroplasts, chloroplasts. Like I was saying, mitochondria and chloroplasts, we study both of them a lot more in the very next unit. But that's a chloroplast. So a chloroplast looks kind of like this. Also sort of looks similar to like a mitochondria. But if I bust it open, instead of having like one big long squiggly membrane, it has these things that look like stacks of cookies or like a sleeve of Ritz or a stack of coins or something like that. So this is a chloroplast. Chloroplast. So plants have chloroplasts. What process do chloroplasts do for the plant? What process do chloroplasts do? If I'm a plant and I've got chloroplasts, what process can I do that humans can't? Yeah, so photosynthesis. I can convert sunlight energy into food energy. They happen to start off by always converting it into glucose. So they have the same meal for every single meal for their entire life. They just make sugar. They never get steak or anything. So they convert sunlight energy into food energy, like glucose. They can then start joining those glucoses together to make sap. And sometimes the sap tastes delicious and sometimes it's gross. So if I stab a maple tree and all the sugar that they've been producing starts oozing out, I can put it on my pancakes and it tastes delicious. It's maple syrup. I can stab a pine tree, and I can put that sap on there too, but it's going to probably taste like pine tree sap, which, I don't know, based on the way it smells, it's probably going to taste pretty gross. So you can put pine tree sap on a baseball bat, and it allows you to grip it better. So if I'm like a Major League Baseball player, you can put like pine tar on your bat. Yeah, so anyway, they convert sunlight energy into food energy. What was that special chemical that they have that allows them to do this? It's almost like a solar panel, like a solar cell. What's the chemical that chloroplasts have that help them do photosynthesis? Yeah, chlorophyll. So chloroplasts have chlorophyll. Chlorophyll. Chlorophyll also has to be stuck in those inner membranes for it to work. We'll see why next unit. It's also got to be stuck in those inner membranes if it's going to work. ATP synthase had to be stuck in this folded up inner membrane to work. Chlorophyll has to be stuck in these membranes to work. All right. In mitochondria, if I want to get a bunch of orange dots into a small space, I take this giant membrane and I just fold it, and now it fits into a small space. With chloroplasts, they're doing the exact same type of thing. It's just slightly different. So now, instead of keeping it as one page, I do this, and I make kind of like a stack of pages. Now I got 4, 8. So now I got like these 8 things stacked. This will still fit in my pocket, and it's still holding the same number of dots. That's what's happening there. I've got like 1, 2, 3, 4. Here I've got like 8 stacked. I'm just stacking stuff like this, like pages in a book. So if I stack stuff like that, I also have a whole lot of extra surface area. Give you an example. If I go back here and grab neuro. So I got this textbook. It's 1,700 pages. 1708. So each page is about 1 square foot. The squares on the floor, those are exactly 1 square foot. Pretend this is 1 square foot. I know it's not exact. 2, 3, 4 square feet, 5, 6, 7. I'd have 1,700 square feet. My first apartment was 600 square feet. It had a kitchen, a living room. The living room was small, and a bedroom. My second apartment had 2 bedrooms. It was 900 square feet. There are houses in the villages of Woodland Springs. The smallest houses in the villages of Woodland Springs are 1,450 square feet. My wife's a real estate broker, so we see all kinds of crap like this. So there's more surface area in this book, 1,700 square feet, than some houses around us that have 3 bedrooms, a kitchen, a couple of bathrooms, and all of that. If I started laying out individual pieces of paper on this floor, I could cover this entire room and still have pages left over because there's more surface area in this book than there is surface area on the floor. Do you see how stacking these like pages is the same as stacking those membranes in a chloroplast? Yeah, so it's the same idea. I can fit more stuff into a small space. So in my backpack, I can have more square footage than an apartment in a book or two. They do give these things names. So this folded inner membrane on mitochondria is called cristae. C-R-I-S-T-A-E. Cristae. So the folded inner membranes in there are called cristae. I don't know why. Christ normally represents Christ. It's from like the root word for Christ. And so if you told me something was named cristae, I'd be expecting something that looked like pluses, crosses, X's, or something like that. So I don't know. No. Somebody just looked at that and they're like, it makes me think of Jesus. And so we're going to call it cristae. I don't know. I don't understand why they would call it that. But they do. So cristae. These little things right here are called thylakoids. Thylakoid. Thylakoid. So the job of cristae being all folded increases surface area so I can hold more ATP synthase. The job of thylakoids being stacked like Ritz crackers is now I have more surface area to hold more chlorophyll. And chlorophyll is the main thing. That's like a solar cell and a solar panel. That's the thing that's doing the work. That's converting sunlight energy into food energy. Right, most of you guys remember this, but there's a tricky question. This is one of the few questions that they can ask on a STAR test and an AP Bio test. Otherwise, there's very little in common between a STAR test and an AP Bio test. So they'll try to trick you. Let's see. Let's say I've got animal cell, animal versus plant. And I have mitochondria and chloroplast. All right. Do animal cells have mitochondria? Sure. A bunch. The average muscle cell has 10,000. Do animal cells have chloroplasts? Nope. Otherwise, world hunger wouldn't be a thing. Just move where it's sunny or roll up your sleeves. Plants. Do plants have chloroplasts? Yes. Do plants have mitochondria? Yes. So they'll try to get you to say that plants don't. And the reason that people would say they don't is they'd be like, well, we have mitochondria. That's how we get energy. Plants have chloroplasts. That's how they get energy. That's not entirely true. We're getting a little bit loose with the word energy because here we're converting energy into another form. So if a plant has chloroplasts, that's how they get food. But do plant cells run on food? No, what do all cells run on almost exclusively? ATP. So they still have to convert their food energy into ATP just like you and I do. We don't have chloroplasts. So what do we do instead? They've got them and we don't. Yeah, we eat. So plants don't eat because they have chloroplasts. That's how they get food. You and I don't have chloroplasts. So how do we get food to convert into ATP? We eat food. They don't have to. They use sunlight to make their food, so that's why they don't eat. So even if I'm like a Venus flytrap, I actually don't have to eat a single fly my entire life and I can survive. I can get all my food from photosynthesis. The main thing that they're using insects for is to get nitrogen. So if they can get nitrogen out of the soil, if you're a Venus flytrap, you can live your long fly trapping life without ever seeing a fly. So they do photosynthesis instead of eating. Kind of makes sense. One last thing on chloroplasts. How many membranes do you see there? 1. And then all the purple ones are, if you're looking at each of these, you're like, I don't know, 1 and then 2, 3, 4, 5. But these are all the same type of membrane. Inner one, that's an outer one. They start looking closely at chloroplasts. They've got their own DNA. They've got their own ribosomes. So we also think that chloroplasts evolved from photosynthetic bacteria. There are currently photosynthetic bacteria. So we think that they evolved from photosynthetic bacteria, kind of like how mitochondria we think came from bacteria also. So again, it's hard to believe, but I can't really come up with a better explanation of why this, if not. So, all right, that's the overarching stuff for organelles. Does it all kind of fit together? I've got ER, vesicles going to Golgi, or I have DNA converted to RNA going to ER, vesicles to Golgi, gets a chemical tag. I need energy for all this, so I make some ATP. If I'm a plant, I make some food. It should all kind of fit together. Below chloroplasts, I mentioned cytoskeleton. You don't need to know the different types. I went ahead and put them in there, but all you have to know about cytoskeleton is its main function. So what would cytoskeleton mainly function to do? It's actually got 2, but there's one that should be obvious. Yeah, holds the shape of the cell. It's the skeleton of the cell. So if I want to hold the shape of the cell like this, I have cytoskeleton, kind of like steel cables or steel beams holding the shape of the cell. Anytime I'm like transporting stuff, another thing that it does is like my kinesins are like walking along pieces of cytoskeleton. So they can also be like roadways. So cytoskeleton is everywhere. If we look inside of a cell and we like stain the cytoskeleton, we make it fluoresce. There you go. There's a nucleus, and all that green, hairy-looking crap is all cytoskeleton. Here's a cell. The nucleus is blue. The cytoskeleton is green. And vesicles that kinesin's carrying along are all that reddish orange. So these are all just vesicles of stuff moving along. I can figure out the shape of the cell even though I can't see the cell membrane just from the cytoskeleton. So this is a nerve cell. Kind of like a house. A house might not even have all the walls up yet, but if they have like 2x4s, I can walk in. I'd be like, all right, this is the front door. I see plumbing right there. This must be the kitchen. Based on the plumbing in the kitchen, this is a sink. Based on the plumbing over there, this looks like this is water for like your ice machine in your refrigerator. Oh, that's where the laundry is. So even if a house is undone, even if the walls aren't up, can I kind of walk through and figure out the basic layout of a house? Same thing, based on the cytoskeleton. I'm like, all right, this cell is shaped like this. All from the cytoskeleton. All right, any big idea questions you have so far? I'm going to go and drop in a few more little details along the way. All right, smooth ER versus rough ER. Did I talk about drug tolerance? Yeah. Let's see, let me go back and see if there's anything that I know that I kind of skipped. Nucleus, got that. We mentioned histones in the nucleus, right? Okay. ER, smooth and rough. Ribosomes, different types. Golgi, lysosomes, peroxisomes, lipid droplets. Cool. The remaining ones, like cilia and flagella, if I'm looking at a cilia or flagella, if I was going to draw, here's a cell, and I was going to draw a cell with a flagellum, what's a flagellum going to look like on that cell? Yeah, just a big long tail. So if you're a female, a biological female, you don't have any flagellated cells. That's a real word, flagellated. If you're a male, the only flagellated cells you have are sperm. Cilia, you don't have cilia on your sperm unless you're a freak. And I don't mean freaky, I mean a freak. Then I didn't make one. I wish I knew how to edit video. So here's a cell with cilia. Cilia work the exact same way. They do the same thing, they're just small. So if it's, you know, one, maybe two, maybe three big long tails, and as those tails move, this thing swims around, we call it a flagellum, or flagella, a bunch of them. If I've got a bunch of teeny tiny little ones and they all move together, we call it cilia. So imagine I've got this cell right here and it's got one big long tail with like one or two whips of the tail. It might like move all the way across here. Whereas cilia is kind of more like, you know, like that. So have you guys ever seen way back in the day, if we had boats and we're trying to get from one place to another, there might be people like down in the bottom part of the ship. There's like hundreds of people like rowing and they're all like rowing together. So if I looked at the boat from above, wouldn't I just see like a bunch of oars sticking out as the boat like very slowly? Like each time they went like row, the boat goes forward like a foot. That's kind of what cilia would look like. Flagella would be like you're a dolphin or a shark with some big powerful tail. So cilia versus flagella. Cilia, by the way, you've got a bunch of cilia on the top of the cells at the top of your trachea. And so I had a professor, Dr. Magruder, told us this when I was in college. He's like, hey, we did this experiment with mice and smoking. And, you know, people have always known, if you've ever like grown up with a smoker or maybe grandpa smokes, both my grandparents on my mom's side, on my dad's side, smoked. So they both died from cancer young. In my family, if you smoke, you die in your early 60s. And if you don't smoke, you make it to at least 85. My Aunt Helen made it to 103. So in my family tree, smoking knocks off about 3 decades of your life. So my grandfather exactly would like wake up and start hacking at night. And he'd hack up this like black tar crap because it was black tar. And so like 2 in the morning, he'd be in there like hacking up this tar. If any of y'all, you don't have to raise your hand if you don't want, but if you know been around somebody like this, like they wake up and they actually hack up like black stuff. Okay, so you'd hack up this stuff, and you're like, okay, I understand when I'm smoking. In the filter, I can see the tar in there, like I can see the brown stuff, so I can see the tar, so I know that some of it's getting in there, but it's making it down to my lungs. So what they did was they had this mouse. It was still alive. It was during a surgery, so to speak. And at the top of your trachea, you have all these ciliated cells. And anytime something touches them, they pulse upward. So if something starts to go down my trachea, they pulse upward, and it goes up. So like right now, if I take a drink and I swallow some water, I close off my trachea to like swallow water. But while I'm not taking a drink, isn't there still like spit, or if I'm having allergy problems, like snot just like draining down my throat all the time? It is. And so if I'm not like purposely taking a drink or taking a bite of food, it's just draining. It's 50-50. Half goes down my esophagus to my stomach, half is going down my trachea towards my lungs. So how do I keep all that fluid out of my lungs? These cilia are always just pulsing upward. So they pulse up. Now there's a 50-50 shot, it goes to my stomach. Pulse up, 50-50 shot, it goes to my stomach. So eventually all of it goes to my stomach pretty much. All right, fine. So what they did was they had this mouse, it was opened up, its trachea was opened up, and they took like a teeny tiny little piece of something. I kind of picture it was like paper or sterile gauze or something like that. They took a teeny tiny little piece of paper and put it at the top of its trachea, and it would push it out. So this animal that was anesthetized, its trachea, the cilia would like push the paper out, push it back in, it would push it out. They call it the mucociliary elevator, the mucus cilia elevator. Then what they did, you take a drag on a cigarette, blow it in the mouse's trachea, see ya? Blow it in the mouse's trachea and put it in there, and it didn't pulse out. It paralyzed the cilia. So cigarette smoke paralyzes the cilia, and so now, thanks for donating blood. And so now, whenever I have all this crap like snot and stuff that's draining down my throat, while I'm smoking cigarettes, it's going into my lungs. And so at 2 in the morning, Grandpa Kenneth would have to get up and just hack out all the crap that he's been swallowing in his lungs all day. He also smoked unfiltered cigarettes. His filters just filter out the taste. That's what he'd say. Sometimes they wouldn't have unfiltered cigarettes. And so he'd get his pack of cigarettes, and the first thing he did, he'd be like ripping off all the filters. So he just went like as raw as you can, as far as just like, I want nothing between me and the tobacco. Wonderful man, but died early because he was a smoker, as we knew that he would. It was sad, but like it wasn't a surprise. It's like, oh, Grandpa Kenneth got lung cancer, and you're like, yeah, of course he did. Like, you knew that's what was going to happen, right? You knew he was going to die from a heart attack due to hardened arteries from smoking, or lung cancer or something. So yeah. All right. As far as prokaryotic cells, if I kind of go a little bit backwards in my notes, basically prokaryotic cells they've got DNA and ribosomes, and that's about it on the inside. Their DNA works the same way as ours. Their ribosomes work the same way as ours. They look slightly different. Rather than that, it works the same way. So yeah, I think we're done. Unit 2 in the books. You guys have like 5 minutes to just chill. I don't think that's happened yet this year.